time, let's get started. Tonight's session will be recorded. Thank you for attending Get to Know Vibrant Salivary Hormones Test, Spotlight on Adrenal Health. I'm Mary Beth Augustine and I lead the clinical team here at Vibrant America. I will act as your moderator, and also joining me are our clinical liaisons, Adair Anderson and Dr. Lily Liu Nguyen. As moderators, we will respond to the live chat and Q&A throughout the presentation. And following the presentation, we've allowed approximately 15 minutes for live Q&A. Attendees are in listening mode, so please submit your questions in the virtual Q&A or chat applications. And now, allow me the pleasure of introducing you to today's presenter, Dr. Deborah Harding, who is the Clinical Lab Education Coordinator here at Vibrant America. Dr. Deborah Harding is a naturopathic doctor, but she is not your naturopathic doctor. The content of this webinar is for educational and informational purposes. The information is not to be interpreted as or mistaken for clinical advice. Please consult a medical professional or healthcare provider for medical advice, diagnosis, or treatment. Dr. Harding has over 20 years of experience in the field of naturopathic medicine. She is a former core faculty member of Bastyr University, San Diego, where she taught integrative oncology, environmental medicine, and clinical medicine. Dr. Harding also worked for many years as a naturopathic oncology provider at Cancer Treatment Centers of America, an integrative cancer hospital in Zion, Illinois. Additionally, Dr. Harding has served patients in general family practice in several integrative medicine clinics, including the Natural Health Clinic and Apothecary that she founded in Marlborough, New Jersey. Dr. Harding is a fellow of the American Board of Naturopathic Oncology. She received her naturopathic degree from National University of Naturopathic Medicine and her BA degree in environmental studies. In her free time, Dr. Harding loves spending time with her family. They enjoy cooking, swimming, and exploring nature. Without further ado, I'll turn it over to Dr. Deborah Harding.
Hello, thank you for that warm introduction, Mary Beth. It's great to be here. Welcome, everybody. Today here we go. Thank you. I'm going to start all over. It's great to have everybody here. Today, I'm going to be speaking about Vibrant America's salivary hormone test with a focus on adrenal health. And this is some preliminary things that Mary Beth already told you. So we'll go on to the next. This live class is categorized into 4 parts. First, there's an overview of the salivary hormone test as a whole. The first part will cover basic analytes on the test, a sample test report, what tests to consider alongside this test, and other general information about the test. The second part of the presentation will cover the adrenal health section of the salivary hormone test. We'll do a bit of a deep dive into how to interpret the basic adrenal panel, and along the way, we'll cover HbA access function and considerations of glucocorticoid testing. And then the class will cover 3 quick questions that often arise with interpretation of the results. And as Mary Beth said, lastly, we'll have some time for Q&A. So, as I mentioned, the first part addresses some introductory information around the test so you can navigate your way around it. Salivary hormone testing strengths are provided in 3 main ways. First, if your patient is suffering with hormone-related symptoms of sex steroid origin, such as hot flashes, night sweats, fibroids, PMS symptoms, PCOS symptoms, low or high testosterone symptoms, salivary testing provides a unique look at the bioavailable hormone concentrations that are active in the tissues. These results may differ slightly from serum or urine levels, as they are an alternative lens to view the hormone picture with. If you're suspecting your patient has glucocorticoid challenges, as a second point, the HbA access dysregulation issues that may sometimes arise with patients, unidentified chronic health issues, the salivary hormone test provides a comprehensive look at adrenal output markers 4 times a day, both cortisol and cortisone. And it also offers DHEAS, which is kind of an adrenal-related hormone, as well as a sex steroid hormone. And thirdly, if the patient is on topical hormones, salivary hormone testing is the preferred method to prevent against overdosages, as salivary testing captures hormone that's not captured through serum or urine. Here's a list of the test analytes on Vibrant's saliva test. And you can see that we offer 3 different estrogens in the salivary sample, which enable us to give an estriol ratio. We also offer a progesterone estradiol ratio in that list. And in our sample, we offer cortisone and cortisol 4 times a day, so we can chart a day to night or diurnal pattern. And here's a look at the actual report. The values are listed out as shown on the left. And on the right is a graph representing the day to night pattern of cortisol and cortisone production. And we'll talk a bit later why viewing this day to night pattern is an added bonus to the test. Some of the tests listed here are for initial consideration and are endocrine diagnostic purposes. Others are here for functional medicine deep dive into patient symptomatology. So if you look at the first one here, you see urine hormone tests. And in this test, the urine hormone test provides metabolites to assess risks of various estrogen metabolism pathways. So how your body breaks down estrogen as well as other hormones. And it gives you a sense of systemic burden as well as some of the types of metabolism pathways that can help navigate extra hormone burden in the person's body. The serum hormones here, estrogen, progesterone, sex hormone binding globulin, can provide luteal phase, ovulation, and other helpful diagnostic information. So the salivary hormone test isn't a standalone endocrine workup. Having serum hormones to assess other items in a person's history to give a full diagnostic view is very helpful. And additional cortisol tests, the same reason, you wouldn't want to do a full diagnosis based on salivary hormone testing because often you need follow-up tests and often diagnostic values are found in serum levels, i.e. this example here is the ACTH stimulation test for primary adrenal insufficiency. And so we do list a few other serum tests here, thyroid panel, chem screen, and those can provide information that help to look at the system as a whole. For example, if estrogen levels are high, which is a common scenario, then these tests can provide liver, gallbladder, and renal tests can provide sort of information about hormone clearance. The thyroid panel can help to assess metabolism in general. These types of things can provide a fuller picture. And on the Vibrant panel also, we list tests that assess the microbiome. So the wheat zoomer and the gut zoomer are helpful to take a look at factors in the gut that can affect hormone levels. For example, on the wheat zoomer, LPS, also known as endotoxin or lipopolysaccharide antibodies, the LPS itself is a large component of the outer membrane from gram-negative bacteria, such as E. coli. And elevations can be linked to dysbiosis, intestinal and blood-brain barrier permeability, and systemic inflammatory effects. And so regarding the gut zoomer, the conjugated estrogens excreted in the bile can be deconjugated by bacterial species in the gut that have beta-glucuronidase and beta-glucosidase activity. So deconjugation, we really don't want happening because it's sort of an uncoupling of the estrogen from the bile, leading to enterohepatic circulation, which increases systemic estrogen levels. So getting a handle on that enzyme activity, for example, the beta-glucuronidase, can help you see, how much of that estrogen may be attributable to a gut source of not having great clearance and instead having recirculation of it. And so this activity is increased by multiple families of gut bacteria, such as Clostridia, Shigella, etc., and they can be overexpressed during dysbiosis. So testing your gut to take a look at the estrobolome microbiome axis can be very helpful. And then just moving through environmental toxins, heavy metals, mycotoxins, these toxicants can interfere with estrogen clearance, also have an added estrogenic effect, and they can negatively impact testosterone production. And lastly, genetic testing through our NutriPro or methylation panel can detect genetic polymorphisms, which may lead to estrogen metabolism issues. Now we're going to turn our attention to the adrenal health indices, which are measured on our tests. And in focusing on the glucocorticoid testing, we are measuring impacts and markers of our stress response system. So what relevance does cortisone and cortisol testing have on clinical outcomes? And this is sort of a big picture question. And this slide gives us the adrenal portion of our salivary hormone test result listed out here. in addition to the types of conditions that are associated with HPA axis maladaptation. And when I look at this as an overall picture, the following is how I approach it. Firstly, are the high and low values for cortisone and cortisol targeted here? Like or put another way, what are the high and low values here? And that's helpful to know. Is one thing trending high, or are many things trending high? Same for low, or are none of them really out of whack? So just looking at a simple high to low breakdown can be helpful. Secondly, each time of day value, as well as the pooled or overall value, offers insight into an aspect of systemic health or lifestyle factors for the patient. So those time of day values on their own, what time is it out of whack, can be helpful. And then third, the shape of the cortisol values plotted on this graph has separate value in terms of what does the curve look like. And we want the curve to have a specific shape, high in the morning and then stepwise down to low at night. And so you could think of adrenal testing for wide-ranging symptoms such as typical thyroid patients or patients with chronic fatigue, depression, or dysglycemic regulation. It offers value if it's positive, and it offers value if it's negative, because a person with chronic fatigue syndrome who has normal cortisone cortisol values can focus less on HPA axis support and pursue another treatment direction. Because who hasn't sort of either thought of adrenal support for a chronic fatigue patient, or have seen them and have them, have 30 different lines of adrenal products in their pantry, so to speak. So knowing if that's an issue or not can help narrow down your therapeutic interventions in hopefully a more effective way. And along those same lines, for treatment, testing these glucocorticoids can offer a targeted treatment that's separate from a condition-specific treatment typically offered to the patient. For example, if we discover that this HPA axis is out of whack, then to a thyroid patient, let's say we have a thyroid patient, hypothyroid, but we see that this is out of whack, we can offer them a separate treatment strategy than simple thyroid support. And depressed patients with irregular HPA axis can also be offered targeted support for this issue, in addition to sort of neurotransmitter type support or lifestyle support. Just broadening our view, our collective understanding of the stress response and the HPA axis largely comes from the pioneering work of Hans Selye beginning in the 1930s and popularized in the 50s. And his theory of general adaptation syndrome describes a model of stress response and adaptation to persistent stress. And it's based on a stressor producing 3 stages of response. Stage 1, initial response to the stressor, is called the alarm stage. Stage 2, the second stage of the stress response, is termed resistance, and that's a heightened stress response. And stage 3 is exhaustion, where that heightened stress response is untenable forever. And then the biological output is depleted. And so one limitation of this model is that in his research, he used rodents. And when they were under stress, they were unable to do anything to stop the persistent stress exposure. And since humans have heightened capabilities of addressing a stressor, more so than rodents in an experiment, the physiological effects from humans throughout these stages would be expected to be somewhat different. Nonetheless, the critical information gleaned from this model, and why we still talk about it, is that it explains a dynamic stress response and hormonal release that changes depending on many factors, and the most pivotal of which is likely to be, how long has the person been managing the stress? And the second question is, to what level has the stress been for how long? That's why this can be helpful as a model. And if we look at the hypothalamus pituitary adrenal axis, it's a commonly known part of our stress response system. And this HPA axis regulates production and secretion of glucocorticoids. It begins when a stressor activates the hypothalamus in the brain. The hypothalamus releases CRH, or corticotropic releasing hormone, which goes to the pituitary gland and signals release of ACTH, adrenocorticotropic hormone. And this travels to the adrenal cortex and stimulates release of glucocorticoids and DHEA, which act as counter-regulatory. The DHEA, rather, acts as a counter-regulatory hormone. And so after the glucocorticoids are released in response to their signals, their presence, their very presence, sends a negative feedback loop to suppress further production of the ACTH and CRH from the hypothalamus and pituitary. So remember this sort of feedback loop. We're going to be revisiting it, although there won't be a quiz. But just keep it in mind, because all that is sort of something that plays into our understanding of HPA axis dysregulation. On the right here, just for your reference, is an overview of hormone production from the adrenal gland. And the adrenal cortex, the outer portion here, secretes corticosteroids, mineralocorticoids, and androgens such as DHEA, while the inner portion of the adrenal gland, the adrenal medulla, secretes our stress hormones, epinephrine and norepinephrine. In addition to the highs and lows of the results, like I mentioned, the actual shape of the curve of the plotted values holds value. And so this is an example of one of those curves. It's the diurnal pattern. An abnormality in the slope of the curve has researched associations with worse outcomes in numerous conditions. And the most commonly studied variation is a flattened or blunted slope, as seen in the salivary cortisone image on the right. You can see that those values are attenuated, they don't produce a normal curve, and they're in many cases low, but not always. To make that flat curve, some may be trending high in the range and some are low, but the overall presentation is a flattened or blunted curve. And this type of pattern with cortisol is seen in patients after a significant period of prolonged stress, like caregiving for a long period of time, early traumatic life experiences, burnout, etc. So you can see there's a dampening of the steroid release throughout the day. And that has researched associations with negative outcomes in conditions as seen in the green box on the left. So your providers are thinking that are just treating depression or cardiovascular disease, but they may also need to offer HPA axis type of interventions to fully support the patient. Especially if you're, I used to work in oncology, and cancer patients sometimes from just the chronic stress of the diagnosis or the treatments, they can too have this flattened curve. And so treating the HPA axis for those patients and uncovering that is a target to treat can be helpful for those patients. Okay, so let's do a case study. So let's begin with a simple study of cortisone and cortisol with this patient's test results. She's a 42-year-old female with a chief complaint of fatigue, and her lifestyle is notable for chronic stress. She's a caregiver for her mother in early stages of dementia. So you can see firstly that the only value that's out of range is the nighttime cortisol. And then you can see it here in the results on the right. You can also see it in the curve on the left, and where you can see that the shape of the curve is mostly intact. And again, it's high in the morning and it steps downward towards its lowest point at bedtime. And she mostly has this structure, but what is notable is that her night cortisol is elevated. So in looking at the overall picture, the first thing to notice is the shape of the curve. And the second thing is the individual time of day of that out of range value. And so for some times of day, the strongest association appears to be situational triggers like hypoglycemia, exercise, insomnia, commuting, drug use, while others can be associated with systemic conditions. And so this late night value that she has in particular can either reflect a situational trigger, but also systemic conditions need to be looked at. And why I say a systemic condition for this one with the late night value is that it reflects the expected nadir of cortisol output and therefore baseline cortisol levels. It should be the lowest point of cortisol in the day. And so if it's not reaching its lowest point, it could reflect some kind of systemic trigger like inflammation or other disease conditions. We'll see more on the next page. And if very elevated, if extremely elevated, it could also indicate a Cushing's disease or Cushingnoid syndrome. And so this value, if very elevated, has evidence-based efficacy towards that. And we would we would see that if, for example, the value were over 5.5 or 6, it should prompt consideration or referral to explore that. We don't tend to see that too often, but it's helpful to know that that can be looked at in that way. So here are some causes of high cortisol values. Some of these you're obviously going to have other things going on with the patient. For example, you see conditions like this in the ICU or life-threatening situations, shock, severe hepatic disease. This is where, the cortisol levels are all very high, as well as Cushing's disease. And then it also is in italics are sort of common short-term normal lifestyle triggers that can cause elevated cortisol values. And then over in this gray box too, you can see drugs that increase cortisol levels, glucocorticoids, caffeine, nicotine, etc. And even marijuana can have an increased cortisol effect from initial use or opposite effects from prolonged use. So there can be lots of lifestyle triggers for these. In the case of our patient, again, we see that her late night cortisol values are elevated. And so for this patient, I would be working not only to sort of say, like, what are her situational triggers? Is she stressed out at night? Is she working late? Is her circadian rhythm off and that she goes to bed, much later than this? And she took this at midnight, but she stayed up till 2 that day. So you could look at situational stress, but also work up for some of those inflammatory conditions or systemic diseases if necessary. And also in terms of our general adaptation syndrome model from Henselier, you're wondering, is this the beginning of a heightened stage 2 stress response pattern where you're starting to see elevated cortisol throughout? And it's hard to tell with a single test. Right now, we just have the one value. Sometimes you can see where the values are sitting in the reference ranges. But for now, repeat testing can help clarify persistent patterns and careful sort of questioning of the patient regarding the variables on the day that she took the test, ruling out some of those lifestyle triggers. Now let's turn our attention to another marker for HPA axis dysregulation that's measured via adrenal output, and that's DHEAS. And so DHEAS is the most abundant circulating steroid hormone produced by the adrenal glands. It's the most sulfated and abundant version of DHEA. And compared to DHEA, it has a longer half-life. It doesn't have that day to night variation, and it provides a stable sort of circulating pool to measure adrenal androgen activity. And by itself, DHEA and DHEAS are just slightly androgenic, but they serve as a precursor to androgens and estrogen in the periphery. And so we also know DHEAS is known to be an active neurosteroid and a buffer from the effects of oxidation and glucocorticoids. And typically, females are referred for this testing for any masculinizing symptoms or PCOS evaluation, and males are referred to this for looking at adrenal tumors or alopecia or primary or secondary adrenal insufficiency. And so DHEAS, like all steroid hormones, begins with conversion of cholesterol into pregnenolone, and then you can see the pathway there. So why I go into that one in particular is it's somewhat helpful to look at it in comparison to cortisol values, because DHEAS acts not only as an anabolic hormone, as we saw on the previous slide, but also appears to have counter-regulatory actions to the effects of cortisol in tissues, such as improving insulin sensitivity, neurological protection, positive immune modulation, and other benefits. And so we know when HPA axis releases cortisol, it also involves concomitant release of DHEA. And this is presumably to moderate the tissue effects of the cortisol. And so generally, DHEA is affected by age, with levels peaking in the early 20s and decreasing sort of predictably in a stepwise fashion throughout the later decades. So if you consider the age of the patient, it can help inform more optimal values in the reference range. Not many labs stratify the reference range by age for the patient, but looking at the overall reference range, you can sort of infer some things. So for example, the DHEAS is at the higher end of the reference range. And so clinically, the art of sort of looking at, from my perspective, it's matching a higher level of cortisol output, which may be modulating it somewhat, i.e. the DHEA is modulating the higher cortisol. In the last slide, I said that DHEAS is produced primarily by the adrenal glands, but it's also produced by gonads and the brain in lesser amounts. And so because the testosterone is not elevated in the reference range, it suggests to me that the adrenal gland is likely to be contributing to this higher than expected value, rather than ovarian production for this patient, because if it were ovarian production, I would also expect the testosterone to be high. So I'm looking at the DHEAS and sort of getting more supporting information for maybe seeing that there's a trend going on here of elevation with that nighttime cortisol. So here's 3 quick questions, and they commonly arise in interpreting the adrenal section of the salivary hormone test. And these questions come up either from the patients or the providers are sort of going over these a lot with folks. So let's give it a try. The questions are, what's the difference between cortisol and cortisone? What does cortisone testing offer clinically? And is the term adrenal fatigue medically accurate? So this is our first question. I'd like to have you guys and our audience type in the chat a few words about what you think the difference between cortisone and cortisol is. And I'm curious how many folks already have a sense to the answer to this question. So don't be shy, just go ahead and throw something in the chat. What's the difference between cortisone and cortisol? Look at you guys, awesome. So I have some participators, thank you so much. We see cortisol. Cortisol is the active form of hormone. And then someone else wrote active versus inactive. Awesome. So you guys are right. As seen on the left, cortisol is the active hormone with strong effects on the receptor sites. And the functions on the left are well known to most practitioners: suppression of inflammation, maintaining glucose and protein homeostasis, mediation of the stress and immune response. And cortisone, as our attendees accurately pointed out, is actually an inactive hormone precursor. And so we see here that 11 beta hydroxysteroid dehydrogenase 2, or called forevermore 11-BHSD2, it converts cortisol to cortisone. And I think of that because I think of number 2 as sort of the lesser one, whereas D1 is the enzyme converting cortisone to cortisol. Sort of 1 is going to the number 1 active one here. No offense to cortisone meant by any of that numbering. But in any event, the conversion to cortisone prevents cortisol from activating mineralocorticoid receptors. So if you can avoid the mineralocorticoid receptors, then you're going to avoid excess symptoms of sodium retention, hypertension, and hypokalemia. And so the body does that all the time, and to a large degree in tissues that require that, especially the kidney. So I just want to point out that cortisone, as we mentioned, is inactive, but in tissues that have a large pool of 11-BHSD1, it's as potent as cortisol because there can be rapid conversion to those to this hormone from the high amount of activated HSD1 enzyme activity. And so this expression is highest in the liver, but also occurs in adipose, skeletal muscle, brain, and other areas that need energy and mobilization quick. Our second question here, what does cortisone testing offer clinically? Anyone have any idea on that one? Please type it in the chat, and I'll give you about 10 seconds. I stumped you. I guess that means it's a good question. All right, any last guesses? All right, well, let's go through it then. No worries, here we go. So this is the point of the lecture, right? So salivary cortisone has been found to be strongly associated with the serum cortisol levels. So has salivary cortisol. So generally, they're both associated to serum cortisol levels. And commonly they trend together. So you should generally see them matching each other. However, cortisone is a slightly better marker of serum-free cortisol than salivary cortisol in situations where serum cortisol levels are low or very high, like during hydrocortisone therapy or acute stress situations. If the cortisol levels are super high or super low, you might want to look at cortisone. Again, especially in someone who's taking any kind of supplemental hydrocortisone. In our patient, because we see the cortisol elevated at night, we see that it's outside the HPA axis curve, but in the cortisol, but we see that in the cortisone curve on the right, it's kind of maintaining the normal curve a little bit. So we could infer a couple things from this. One is we can infer that it's a good sign that the reserve of inactive cortisone is functioning in a normal pattern, i.e. this is more functionally optimal than if the cortisone were also elevated and out of range or out of whack, so to speak. However, the discrepancy could reflect something else. Always check to see if the patient's taking exogenous hydrocortisone, as this would cause an increase in just the cortisol. And in these cases, you could give more weight to the cortisone values if they differ. Also consider the activity of that enzyme, the 11B-HSD1 function. And just another word about that. As mentioned, these two enzymes can affect cortisone and cortisol levels, and they typically affect local target tissues in an integrin manner, as in 11-beta-HSD2 upregulates in the distal nephron, and 11-beta-HSD1 upregulates in adipose tissue in the brain. And in these scenarios, the effect is integrin, i.e. it's local, and it doesn't affect systemic cortisol production. However, these enzymes can also work in an endocrine manner, and they may affect the levels systemically too. And for example, 11-B-HSD1 in the splanchnic bed generates about 30 to 40% of the total daily production of cortisol in humans, while 11-beta-HSD2 deactivates a similar percentage in the kidney. So up or down regulation of these enzymes can play a role in systemic cortisol and cortisone dynamic levels. So what kinds of things affect these? Well, chronic diseases, metabolic issues can affect them, but also drugs, pollutants, hormones, and foods can even affect upregulation and downregulation of them. And so if this is the situation where you think you might benefit from knowing more about these specific enzymes based on your patient's test results, then it's a good idea to consider urine metabolite testing, because that will give values for the metabolites of each of the cortisone and cortisol and help clarify systemic rather than salivary output. And that each of those metabolic pathways can help you infer which enzyme here, the 11B1 or 2, is more systemically dominant. Last quiz question. Is it medically accurate to say that adrenal glands get fatigued from chronic stress? Go ahead and write your answer in the chat. Is it medically accurate to say adrenal glands get fatigued from chronic stress? All right, thank you, Dawn. Everyone said no. Fantastic. Everyone already knows, but I'll go through it anyway. That's great. So it's really a question of nomenclature of the term adrenal fatigue. A few decades ago, the term adrenal fatigue was groundbreaking. It popularized the idea that chronic stress has effects on adrenal output and that these effects are measurable. And it created a new therapeutic target and quasi-diagnosis for symptomatic patients with suboptimal levels that were positioned between the extremes of Addison's disease and Cushing's disease. However, what has come to consciousness after years of telling people they have adrenal fatigue is that the mechanism of chronic stress and altered adrenal output don't appear to have a lot to do with the ability of the adrenal gland to produce hormone, not in regard to clinical situations between the extremes of adrenal insufficiency and Cushing's. There's no evidence that we can find that the adrenal gland is compromised in its ability to manufacture hormone at these sort of suboptimal levels of output. So typically when levels are low and adrenal insufficiency is ruled out, decreased output of cortisol and DHEAS are commonly found to be from HPA axis downregulation or something called glucocorticoid resistance as an adaptive strategy to protect cells against persistently elevated levels of cortisol. And so bringing it back to our HPA axis picture, many of the dysfunctions related to chronic stressors are related to a disruption in the feedback mechanisms, whether it's a insensitivity or a sensitivity, it tends to be here in the feedback and in the receptors that the problem is theorized to lie. So just looking again at our at the issue of low cortisol values, outside of primary and secondary adrenal insufficiency, other causes of hypocortisolism relate to and are informed by systemic non-adrenal triggers: inflammatory, chronic stress, infectious, other glands, drugs, etc. Corticosteroid use, either long-term or recent withdrawal from, is the culprit commonly with low output. And any form of repeated long-term stress, either physical from an infectious process or psychological from early traumatic experiences and chronic burnout from caregiving, can also result in low output. And this low output is a result of an HPA axis maladaptation, as we mentioned. So thoughts on HPA axis dysregulation for our treatment approach. You know, as we can see here, there's no shortage of adrenal support products to choose from. You know, we have cortisol dampening nutraceutical products, cortisol enhancing products, cofactors for steroid hormone production, hormone replacement, or glandulars. Keep in mind that dysregulated cortisol production can be triggered by a plethora of non-adrenal factors, and treatment addressed towards systemic causes on the HPA axis dysregulation would likely be more effective than standalone adrenal quote-unquote products. And we know that low-grade chronic inflammation underlies most chronic conditions, for an example. And we see here that that mechanism of low-grade chronic inflammation can upregulate 11-beta HSD1 and increase local cortisol levels, which can then sometimes, if they get high enough, spill over into systemic circulation. And then other non-adrenal issues: circadian rhythm disruptions, dysglycemic states, excessive exercise, insomnia, drug use, etc. Let us know that we should try to treat the cause. Okay, so that brings us to the end of my slides. We ended a little bit early, so we have time for questions and answers. Thank you for your attention so far.
Thank you, Dr. Harding, for an excellent spotlight on adrenal health. I did see 2 questions come in. If you want to give those an answer right now. The first one is, what is the best time in the menstrual cycle to collect a saliva sample? And then the second one is, what are catecholamines?
Okay, great. Oh, thank you, Mary Beth. Yeah, so the first one, the best time to collect a salivary sample is during the luteal phase, and typically day 19 to 21 are the best days if someone's menstruating. We have a well-established luteal phase range for these. But if the person's menopausal, we have a reference range for that. If the person is male, we have a reference range for that. If a person is in the follicular phase and they just want to test during that time, we also have a reference range for that. So generally our default is to go for luteal phase. But if there's also some irregularity, we have special instructions for that too.
Thank you for that answer.
Yeah.
And we have a few other questions coming in. If you wanted to address the what are catecholamines?
Yeah, sure. So catecholamines are hormones that function as neurotransmitters. I mentioned them in the Hans-Selye slide that the adrenal glands produce. They produce not only glucocorticoids in response to an alarm phase, but also catecholamines. So your main catecholamines would be things like dopamine, norepinephrine, and adrenaline, and they all have a role to play in how we react to stress, with producing a fight or flight response, increasing oxygenation, increasing chem neurotransmitters to help move muscles for the skeletal system, increasing heart rate and blood pressure, those types of things, so a person can adequately respond to a stressor. Great. I think I see some more questions coming in. One from Janet. What does the vibrant adrenal test measure? 1 saliva sample, 2 saliva samples? Well, that's a great idea. So to talk about that. Yeah, so when you get the saliva kit in the mail, you'll get 4 tubes. And so generally you'll get 4 times a day for those cortisol measures. You'll be taking the test. Usually that's, morning, lunch, evening, and bedtime. And so to get the hormones, they generally pool all of them together. to get the hormones, the sex steroid hormones, estrogen, progesterone, etc. And so they also do pooled cortisone and cortisol, as well as those 4 times a day. They do those first and then they pool the rest for the hormones. Hopefully that answers that one. Do topical body products affect the hormone values in salivary hormone test, i.e. if they contain endocrine disruptors? That's a great question. Yeah, so if in our urine hormone test, we offer a BPA test, which is sort of an endocrine disruptor. We don't have that in the salivary. So if you're hoping to get a value for that, we do have it in the urine hormone test. We also have other phthalates and endocrine disruptors in our environmental toxicant test. So that's one way to measure endocrine disruptors. But in terms of like, are there phthalates that are gonna, I guess your question is it gonna raise estrogen, and then your estrogen is gonna be measured in the salivary hormone? And I think it's a really great question. But I would say at this time, we don't know for sure that it actually is, we're measuring endocrine disruption when we measure estrogen. So what we are seeing is actual E1, E2, E3. And what those tend to do is they tend to stimulate receptor function to have an estrogenic effect. I think whether they actually increase estrogen is a different question. But we know for sure that they have an estrogenic effect, which has issues down the road for estrogenic conditions, you know, in terms of fibroids or cancer or whatnot. So hopefully that addresses that one. Yeah, I think Janet is asking about the test and the cost. I don't know if any of our Adair or Dr. Lilly have information handy. Do you guys have that handy now, or do you want to get back to Janet later with that one?
Dr. Harding, I've asked Janet to contact her sales manager for pricing information.
Oh, great. Thank you. Is there a fibroid hormone guide? Oh my goodness, what a great question. We just published the salivary hormone interpretive guide today. So as of today, that is now in the portal. And so that is a great resource. So enjoy it. It's brand new, hot off the press, right there in the portal as of today.
And Dr. Harding is being modest because she's the author of that interpretive guide.
Well, thank you, Mary Beth. All right, so there she is, Mary Beth. All right, she already wrote that down there. Okay, does anyone have any other questions that are coming to mind on this presentation?
Did you address I may not have seen it, there was a question about topical body products.
Yes, that's when I started talking about endocrine disruptors and phthalates and BPA and whatnot.
My apology, I was responding to someone else.
No worries. Thank you. All right, great. Well, if there are no other questions, I just want to thank everyone so much for coming. Let us know if this test is working for you and your patients. I hope that it does. I've seen a lot of help in this test, and I'm excited about potentially extending its use for you and your patients. Oh, wait, I see a few more questions. Hold on. But wait, there's more. Hold on. Can I explain why salivary hormone testing is better at measuring hormone replacement retesting than urinary hormones? Yeah, I wouldn't say that they're necessarily better. I would say that they measure different things. For example, if you look at urinary hormones, it's measuring metabolites, which can measure things like metabolism and pathways. A very great benefit of that test is estrogen excess, and you're wanting to see if it's going down some pathways that are associated with more harm or more benign estrogen metabolism pathways. And so that can be a good adjunct to this test in terms of if you get elevated estradiol on this test and you want to see, well, geez, my patient has fears about breast cancer, or maybe she is a breast cancer survivor and she's alarmed by this extra estradiol. She wants to see what further metabolism products can show her. Then the urine test would be a better test for that. In terms of measuring hormone replacement, they each have their pluses and their minuses. What the salivary test is really great at is just making sure their patient's not overly being overly dosed on topicals, because sometimes the topicals don't show as well in serum or urine. And so they do tend to show up in saliva. So I think this is one unique thing about the salivary hormone test. And so they all have a slightly different view in terms of what type of HRT is the patient on. There's so many types, and then what might be the best test, serum, urine, saliva, to get to monitor it. But yeah, I would tend to steer someone towards this one if they're on topicals. Okay, great. You're welcome. Well, thanks, guys. It's been a pleasure. I hope you have a great night, and we'll see you again soon.